Welcome back to O-Level Oasis, your ultimate guide to CAIE exams. We're currently exploring the O-Level Biology 5090 syllabus here, and so far, we've covered chapters 1 to 4, along with the first unit of chapter 5 on this channel. Today, we're moving on to the next part of chapter 5, which is enzymes. Specifically, we'll be diving into unit 5.2, which focuses on the effects of temperature and pH on enzyme activity. As always, I've attached the notes for this chapter, just like the previous ones, in the description below and in the pinned comment, so don't forget to check them out. So, let's get started. And here are the key learning objectives we'll be covering in this section. Specification number 1 states, understand that the progress of enzyme catalyzed reactions can be monitored by measuring the concentrations of reactants and products. In our previous unit, we learned that catalysts speed up chemical reactions without being used up. Enzymes act as catalysts by lowering the activation energy needed to start a reaction. Now we will learn that enzyme activity can be tracked by measuring changes in reactant and product concentrations over time, allowing us to observe reaction rates and how they vary under different conditions. Understanding how to track the progress of enzyme catalyzed reactions is a key concept that frequently appears in both practical work and exams. There are two main ways to monitor these reactions. One is by measuring how quickly the product forms, and the other is by measuring how quickly the substrate disappears. Both approaches help us understand the rate of the reaction as it moves forward. A common method involves taking samples from the reaction mixture at regular time intervals. We then test these samples to find out the concentration of either the reactants or the products at each time point. This gives us a clear picture of how the reaction changes over time. A good example of this is the reaction involving catalase, an enzyme found in potatoes. Catalase helps break down hydrogen peroxide into water and oxygen gas. In this case, we focus on the oxygen gas being produced, which we can measure to see how active the enzyme is. To carry out this experiment, we place pieces of potato, our source of catalase, into a conical flask along with hydrogen peroxide. The flask is sealed with a bung and connected to a delivery tube. This tube leads into an inverted measuring cylinder or a gas syringe, which is placed in a trough of water to make sure no air gets in. As the reaction happens, we see bubbles of oxygen forming and collecting in the measuring device. By recording the volume of oxygen produced over time, we can work out the rate of the reaction, giving us useful information about how efficiently the enzyme is working under different conditions. This is a simple yet effective way to study enzyme activity, and it also helps us understand how factors like temperature, pH, or substrate concentration can influence the speed of enzyme-catalyzed reactions.